and wanted to try to think about the different stages that he went through when he was creating, whether it was a mathematical equation or thinking of poetry or something like that. What are the different stages? It's not just this one mysterious thing. There's different subcomponents of creativity that we can start to parse out. The first one is preparation. You have to have stuff in your brain in order to be creative with. You have to have the raw materials of creativity. The artist has to understand something about color and tone and hue and perspective and maybe the human figure to be creative. And that takes some time. That's the 10 years, 10,000 hours thing. You have to practice at your craft in order to be creative. That's the preparation. You have to have those raw materials. We talk about children being creative. Well, they're producing novelty. It's not particularly useful because they don't have that preparation going in. They're good novelty generators, but they're not good creators. Their parents love what they're doing and put it up on the refrigerator. It's good novelty generation. It's original, not particularly useful. And I'm going to be demonstrating some of the techniques that we have throughout. So this is my brain, one to prove that I do have a brain in my skull, and the other to show you some of the techniques that we use. Because that movie's advanced, I want to start it over again. This is a technique called structural magnetic resonance imaging. I don't tend to do functional imaging techniques. And I like structural techniques. It's the experiences that have been laid down in your brain over long periods of time. So one of the techniques we use looks at the cortical thickness at each different part of your brain. So you have your occipital lobe, parietal lobes, temporal lobes, frontal lobes. We're able to measure the cortical thickness from 1 to 3, 4, 5 millimeters at each individual section of the brain. And as you use your brain, when now learning, the different parts of your brain beef up like a muscle. If you're using it more consistently in a certain way, you'll lay down more tissue as an adaptive mechanism to meet environmental demands. So the tissue in this particular part of the brain may be more beefed up in association with auditory processing if it's in the temporal lobe, or spatial or math processing if it's up here in the parietal lobe. You can see there's not a lot of areas that are beefed up in my brain. I'm using it for lots of different things, and I don't know if I have any specific beefed up capacities. But some people, they've done studies, like the famous juggling study by Gyrus. They had people, they measured their brain volume. They were not jugglers. They taught them to juggle for 3 months, and they became expert at juggling. They measured their brain. There's a particular area in the temporoparietal junction right about here in the right hemisphere, and they saw this beef up, lay down tissue associated with learning how to juggle. They stopped juggling, and that tissue started to actively back down to its original volume level. So this is one of the techniques that we use to see where in the brain, you can ask the question, where in the brain is creativity associated with thicker or thinner cortical volume in the brain. So skill acquisition, polymath, we talked about 10,000 hours. These are some of the things that are important to preparation. Incubation is critically important to the creative process. Dr. Simonton talked about that. This is one of the things that I think we're studying most carefully and rigorously. Rigorous is too rigorous of a word. It's persistently studying incubation, or trying to study incubation in this field. And I'll talk about some of the weaknesses towards the end of this talk. One of the techniques we use is diffusion tensor imaging. This is the white matter of my brain traversing the corpus callosum. And I'll take a seat here and articulate different regions of connectivity linking together the spinal column and regions within the parietal and temporal lobe, more local connectivity in the language areas of the brain, more diffuse connectivity, linking up different networks of the brain. We're starting to get an appreciation of the interconnectivity of these different networks of the brain and how important that is, that it's not just one region of the brain that's going to be important to creativity. It's going to be a network of different regions working in harmony that draw creative ideas together. And diffusion tensor imaging is one of the techniques looking at water diffusing through these tubes in the brain. The myelinated axons, it's analogous to the blue wires that connect your computer to the internet, right? So your computer is a very powerful processor, and it's like the processors on the surface of your brain, the cortical mantle. We measure the thickness of the cortical mantle, very powerful processors. But When you connect your computer up to the internet, it makes it that much more powerful. You can pull information from other computers to your computer to do local processing. That is analogous to how the brain works with these wires, these myelinated axons, pulling information in from different nodes of the brain that makes the processing that much more powerful. And we're getting greater appreciation of these networks of the brain that are working in harmony to do cognitive function. So this is one of the silly ways that we measure incubation. It's called divergent thinking. So we ask someone, tell me as many different ways you can think of to use a brick. So, well, you can use a brick as a paperweight, you can use it as a doorstop, you can use it as a mock coffin at a Barbie funeral. That one's different. To throw through a window, to use as a weapon to hit my sister on the head. So you can see there's, and again, Dr. Simonton talked about the fluency of ideas, the flexibility, the frequency, and the elaboration. So fluency is the sheer number of ideas. There's what, six ideas here. Some people have 16 or 2. That's the fluency. The flexibility, number of different categories. So there's some of them are used as a weight, a decorative item, a weapon, a weapon, a weapon. There's a problem. Might be a problem with this person. So we have 3 weapons, a decorative item, and 2 weights. So we're getting a theme here from a psychological perspective. Originality is the one that we're really interested in. Is there something that is really original compared to the other responses? A mock coffin at a Barbie funeral, very low probability item. It's original, yes. Is it useful? Maybe not. So we have to balance those again from that perspective. Another way that we measure divergent thinking is asking these kind of open-ended questions. What if people could change back and forth from boy to girl whenever they wanted to? What would be the implications for that? Have people write about the implications for that. Fluency, flexibility, originality, elaboration. These are the different ways that we can measure these. And then using a consensual assessment technique, getting peers to evaluate their response, we can rate them on a scale of 1 to 5, let's say 1 being least creative, original, 5 being most original. Is this not creative? Is this highly creative? It's not the best techniques in the world that we have, but they're what we have. Illumination, this aha moment, the flash of insight that people talk about. I don't think it's critical to creativity to have this, but some people describe this and some people measure this through the remote associates test, as you've heard about. This flash of insight that people talk about. There's this eureka moment Archimedes we talked about. Mark Beeman talks about this aha moment that he's localized to different regions of the brain, bringing the creative idea from the unconscious to conscious part of cognition. There's this unconscious kind of churning of the brain where things are not in our conscious awareness, but ideas are molding and melding and percolating, bumping one into another in this blind variation type of activity. And then when they bubble up into conscious awareness, you can selectively retain the good ones to push forward. A test of this is a remote associates test. It's not a great test again, but you have 3 target words. You have to think of one word that binds them all together into compound words. So you have lake, mine, shaker. Salt is the answer. Salt lake, salt mine, salt shaker is the one word. And people don't usually arrive at the answer because you heard how quickly that happens. It doesn't usually arrive through a process of elimination or trying different words. It just springs into your mind all at once. This is an aha kind of instance or insight type of process. That's what is believed that is happening with this. And just to demonstrate, this is magnetoencephalography, a technique that we use to study the magnetic impulses of the brain. I'm going to have to start this over too to show you the complexity. A lot of people have seen fMRI, functional magnetic resonance imaging. And usually, by the time you've seen these studies, there's one bright blob or 2 bright blobs in the brain that show this is the region for romantic love. This is the region that makes me love 30-year-old scotch. This is the region that is moral reasoning. It's not the way it works. This is the region that is active in your brain 100 milliseconds after stimulus comes on the screen. It's called the P100. Your your brain lights up like a Christmas tree. Your whole occipital lobe is lighting up. This is how the brain actually looks. It's fascinating. Your occipital lobe just lights up like wildfire, and the dorsal and ventral propagation starts to happen afterwards. A couple hundred milliseconds down. It's 100 milliseconds, 1/10 of a second of activity, just when the stimulus goes on the screen, the P100. And you can imagine the complexity of the thought processes that are going on in the rest of the brain. I have other movies about this using magnetoencephalography. There's a lot of down selection that goes on with fMRI and imaging. This is one of the reasons that I use structural imaging. It's a lot simpler than functional magnetic resonance imaging, which requires a lot of down selecting. Finally, verification. You have to go back to basics. This is a standard MRI of my brain, slicing through front to back, showing all my glitches and foibles in my brain and things that anyone who knows their brains might see some errors in there. But this is where personality variables, the selective retention part of creativity comes into. Personality variables will probably be important with openness to new experiences, grit, determination, persistence. We have to push ideas out into a world that doesn't want to hear all your great ideas, right? They like doing things the way they've done them in the past. So this great creative idea is going to take some getting used to. So, where in the brain is creativity? We embarked on this study with an open mind, completely open mind, as now I've shown you. And we really did not have very well laid out hypotheses. This is an exploratory study, is what we call these in the neurosciences. So the brain is our territory, and we're going on a journey to see where creativity might be correlated with these different measures. The first study, we used a technique called magnetic resonance spectroscopy. It allows us to look at the chemistry, the biochemistry in the brain using a standard MRI machine. This is one of the first techniques actually preceding structural magnetic resonance imaging. And there's only certain chemicals that you can see in the brain that have protons, hydrogen atoms, as part of their chemical makeup. So because MRI machines were basically big bags of water, water is made of H2O, the H, the hydrogen atom, the protons are what we're actually manipulating with the magnetic frequencies, and the big magnet to have them align with or against the magnetic field, and we're perturbing those magnetic atoms in such a way that we're able to create signal. That's about as complex as I'm going to get. We can look, if molecules have hydrogen atoms, protons as part of their makeup, we can actually see those. They resonate slightly off frequency from the water molecule, but we can see them, certain atoms in the brain or in any tissue. Some of the molecules or the atoms and the biochemicals that we can see are very interesting and important. One of them, N-acetyl-aspartate, NAA, is found only in neurons. That's kind of cool. Myoinositol is found predominantly in glial cells. So you have a neural marker and a glial marker. Choline is a measure of tissue cellularity. That's important for tumors and cells that are multiplying at high cellularity. Creatine is a measure of tissue energetics. So you can see that there's only a few chemicals that we can see, but they're very important. So we asked a question: where in the brain is NAA a measure of neuronal integrity related to creativity? Now, for a lot of reasons, we can only look at certain regions of the brain, so there's this big slab where we can look at different squares, voxels of tissue. And long story short, we found a region in the frontal lobe called the anterior cingulate cortex, where NAA, N-acetyl-aspartate, a measure of neuronal integrity, was related to creative cognition, these measures of divergent thinking. Now, the relationship was surprising because it was inverse in our average college-age students, average intelligence. Higher NAA, lower creativity. Lower NAA, higher creativity. It was as if the anterior cingulate gyrus or cortex, the integrity of this, was gating the information in such a way that more information was leaking into the frontal lobes and spreading more widely in those individuals with higher creativity. This is a disinhibitory model of creativity. Very interesting. The second question, we looked at our structural imaging data. We asked the question, where is the cortical thickness related to higher or lower creativity? And there was a region in the frontal lobe where this measure of creative achievement, the creative achievement questionnaire, was related to higher creativity. But again, because it's colored blue, as you can guess, it was an inverse relationship. Lower cortical thickness, higher creative achievement. Again, this is the orbitofrontal cortex. This is a disinhibitory kind of model of fewer brakes on the system, fewer checks and balances on your behavior, higher creative output in the world. Again, fascinating. Finally, we looked at the white matter, the wires of the brain, the integrity of these different tubes that are transmitting water, transporting water, and we can look at the integrity of the myelinated axon, the wires that are connecting up different regions of the brain. Ask the question, where in the brain is the integrity of these wires correlated with creativity? Region in the frontal lobe, this is an axial view. Left, this is radiological convention, so left is right and right is left. So I do know that this is right to you, but it's actually the left part of the brain, left frontal lobe. The anterior thalamic radiation was correlated with higher creativity. The relationship was inverse. Lower integrity of these white matter attracts higher creativity. This is really starting to coalesce into a story of slightly decreased integrity of the frontal lobes, leading to higher creative capacity or output on these measures of divergent thinking. Now, these are normal college students. This isn't an impaired cohort. These are dozens and dozens of college students. Well, slightly impaired, at least on weekends, certainly. But not neurologically or psychiatrically impaired. These are normal college students. So we looked at the entire literature, structural literature, to see if the story held. And sure enough, there's stories out there. I mean, Miller, Bruce Miller, I believe, at the University of California, San Francisco, had done studies of temporal lobe dementia and found that as people progressed in their temporal lobe disease, their frontotemporal dementia, and particularly temporal variant of frontal lobe dementia, in certain cases their creativity flourished and bloomed in these particular case reports. Others did lesion studies, people that had traumatic brain injuries or strokes, and found that in particular regions of the brain, in the thalamus and in the frontal lobes, medial frontal lobes, if you lesion that, if you damage that particular region of the brain, their creative capacity increased ever so slightly in this patient cohort. It's not universal. There's some regions in the brain, the dorsolateral prefrontal cortex, where we and others found increased cortical thickness, increased integrity associated with increased capacity. But you see a lot of these blue regions as opposed to the red. That story generally conforms to this disinhibitory network. And it even more corresponds to a study that I found fascinating of rap musicians and this fMRI study. And they had these musicians doing improvisational rap in the scanner. And you see a downregulation of dorsolateral regions of the dorsal and lateral, the outside rind of the brain, and upregulation of medial regions of the brain. It was a fascinating study that really got me thinking about these different networks, because these networks, the precuneus and the medial frontal cortex, correspond to the so-called default mode network. And we're seeing a lot of overlap between regions of blue in my study and regions that overlap with the default mode network. Could it be that the default mode network that has recently caught fire and interest in people's minds in the neurosciences had something to do with creative cognition? Here's an example of one of the participants and one of the co-authors in this study. So I thought it was cool that he both was a participant and a co-author on this study. So it's really a nice study. So, kind of giving you a fire hose of information, but there's two very distinct networks of the brain that I think are important to this discussion. And I think one corresponds to this internal thought network. The default mode network is when we're thinking about our own problems, when we're thinking about the future, when we're thinking about our relationships, when when we're thinking about problems that we might want to solve. We're not solving problems out in the world. We're kind of cogitating on things. That's this internal thought network, this default mode network. And there's a lot of correspondence between the medial prefrontal cortex, the precuneus, as I say, regions in the anterior temporal lobe, remember that study of frontotemporal dementia, and regions that we see in our studies of creativity. And this is also interesting in terms of research that is done in evolutionary psychology about different reasoning processes. Are there two different reasoning processes? One that is more general purpose problem solving with situation-specific improvisation. It's involved in inductive and abductive reasoning as opposed to, and this heuristics that Dr. Simonton was talking about. It's important to have approximations about reasoning and problem solving before rules are in place. And this simulation of problem solving is also a great time saver and a great evolutionary creation. We don't have to try every idea out in the world. We can run simulations in our mind's eye before we put them into the wet world, the messy world out there. This preparation and incubation, I think, is an important overlap with this mental simulation, this internal thought network, this default mode network. Now this is in contrast to the external thought network with the cognitive control network, the attention network. This is the dorsolateral prefrontal cortex, the superior parietal cortex. We've done previous research that sees significant overlap with intelligence networks, and this is bigger, better, stronger, faster networks in the brain. This is special purpose problem solving, rule-based, deductive reasoning. It's very fast and very efficient, but it's very limited in its applicability. If you think about deductive reasoning, A plus B equals C, it's a very specific rule for a very specific situation. It works very well, but it works in that situation alone. So deductive reasoning, cognition in the external world, I think this has something important to do with illumination and verification. So it has something important to do with creativity as well, but it's that pushing the idea out in the world part of creativity, not the incubation, not the cogitation part of creativity. And it's this back and forth between novelty and usefulness, between internal and external thought processes, between the default mode network and the cognitive control network that I think research will be focused on in the future to understand the neural networks, the neural underpinnings of creativity. I think this is viable going forward, in my not so humble opinion. So, let me skip that. The way forward. I think that creativity is a complex cognitive construct. It will be critically important to fractionate these things. You know, instead of having creativity be this monolithic, insoluble thing, you're starting to see people fractionate it. Even Dr. Simonton was fractionating a novel and useful into 3 parts and increasingly tractable parts. Novelty versus useful. Now we need to fractionate novelty into originality versus surprise, unconscious versus conscious processes, incubation versus verification, simulation versus action, default mode network versus cognitive control network. This makes it a more tractable problem to the cognitive neurosciences. And we should use reliable and valid measures. Our behavioral measures are way too focused on measures of divergent thinking. This concept of blind variation and selective retention is hugely important to the field of creativity. And the Creative Achievement Questionnaire gets as close as we possibly can to that notion of blind variation and selective retention out in the real world. Improvisation holds great promise in terms of understanding how the brain works in that incubation and creating things on the fly and downregulation of one brain network as you're upregulating another one simultaneously. This flipping back and forth between different brain networks, I think, is very important. And triangulation is key with respect to neuroimaging. Over-reliance on fMRI is always a bad idea, in my personal opinion. So lesion studies, structural magnetic imaging, diffusion tensor imaging, magnetic resonance spectroscopic imaging, fMRI, EEG, all of these, if we can pull them together, we'll have a better understanding of the networks that underlie creativity, as opposed to being that person that was stuck with an elephant and touching the trunk and thinking it's a rope, and the ear and thinking it's a palm frond. You have to get a full picture of this very complex entity, cognitive construct. And brain networks hold out greater hope for understanding this as opposed to bits of brain. We've had kind of a phrenological approach to understanding brain with this advent of fMRI, and this bit of brain does this, and that bit of brain does that. Now we're starting to pull the fMRI studies and other studies together into a network perspective of brain. I think that's a very healthy maturing of the field, and we're hopefully part of that growth process. Finally, I want to brag on a student of mine, Sephar Ryan, who's doing some work using graph analysis. And graph analysis allows you to take any type of information and feed it into this graph. And we're using diffusion tensor imaging data and feeding it into this graph. And it can correlate I to J and look at the cross correlation of different nodes in the brain and look at the connectivity of these and the strength of connection in these different networks. We can parse the brain using a technique like FreeSurfer, which allows us to look at different meaningful parcellations of the brain, create these different nodes that we can look at the connectivity of different regions of the brain, structural connectivity. We have a diffusion tensor, we feed that in and we get a graph. And we have some very interesting initial results that are under review currently, where we found some differences between males and females on our creativity measure in terms of the connectivity. We have a negative relationship between composite creativity index and connectivity in the dorsolateral prefrontal cortex in females, but a positive relationship between efficiency in the dorsolateral prefrontal cortex in males. I don't have any idea what that means, but I'm sure we told a good story for the reviewers. It's fascinating that we can look at different connectivity, and there's this old saw about females having more connectivity and males having more local connectivity. We're seeing kind of a different process related to creativity. This is our composite creative index, where males have higher connectivity and positive connectivity in long connections between frontal and parietal cortex related to their performance on creativity measures. And females have more local and frontal disconnectivity related to their performance, if you will, on creativity measures. It's very interesting. You can imagine feeding children versus adults, schizophrenic patients versus healthy controls, lots of different types of methods or arrangements that we can look at with this data. So I think it's young days. Don't be afraid. Creativity is a viable area of research. I know it would be wonderful to study C. elegans, Drosophila, and stuff like that to cure a disease, but I think this is an area of research that is well worth your interest and attention. Thank you for your attention. I'm happy to take some questions. Yes, sir.
Outside of play, beer, and
I can't hear a word you're saying.
outside of